All right.
Thanks, Tara.
Cool. Thanks for having me. That was an interesting experience. I do love the general meetings of committees. Thanks for having me though. So yes, as Sabine said, I'm currently at the Australian Museum. I'm a scientific officer there. I only really just started full-time a couple of weeks ago under a linkage grant through the ARC. And I'll talk about that work today, but I did sort of start getting involved while I was part-time at the museum in 2020 through to the end of 2021. So anyway, here we go. So today I'm going to be talking about essentially exploring fossilization pathways in an iron-rich Miocene rainforest lake Lagerstätte, so an exceptionally well-preserved deposit from central New South Wales. And a little bit of citizen science. I'm just going to throw that in. So first of all, I'd also like to acknowledge the traditional owners of country throughout Australia and recognise their continuing connection to lands, waters and communities. And I'd like to pay my respects to the Aboriginal and Torres Strait Islander cultures and to elders past and present. So about 5 years ago, there was a farmer and he was ploughing his field. This is in central New South Wales near Goolgong. And he came across these rusty red rocks with impressions of leaves on them. Now, it just so happened that this site that he sort of stumbled across is actually quite close to the Talbragar Jurassic fossil fish bed, which contains fish, plants, and insect fossils. And it's actually quite well known. It's been studied for over 100 years. It was first studied by Robert Etheridge, who was actually the first paleontology curator at the Australian Museum. So you can see here, there's a publication date of 1895. And specimens from this site, so they're very well preserved, are in museums and galleries across the world. And until recently, it was the only fossil fish site in New South Wales. Now, more recently, paleontologists have been excavating this site since probably about 2011, mainly volunteers, so sort of expert fossil hunters. One of them knew the farmer, and they'd been going over there and sort of excavating this material. But a lot of it had been studied, a lot of it had been documented and published for quite some time. Now, the farmer said, hey, so he knew one of the volunteers, he said, hey, I've found these fossils over on the other side of the hill. How about you come check these out? So at first they started visiting this new fossil site while they were still focusing on Talbragar. So they were still going to Talbragar, digging up the fossils from there. And as they were walking through the field, they'd kind of, you know, look through some of these exposed rocks that were at the surface. But quickly, it became quite clear that this new site contained exceptionally well-preserved fossils. So this is one of the first fossils that was sort of picked up at the site. This is a perigid sawfly. You can see here it's beautifully articulated. Most of its, you know, bits and pieces are quite clear. Well, relatively speaking, the thing is pretty red, but this is all goethite, and I'll get into that in a minute. And this was later described as a new species of perigid sawfly in the fossil record. They also found a fossilized feather of a small bird. So this is quite small. But it's beautifully preserved. These finds turned their attention away from Talbragar and they started digging up the fossils at this new site. So first they started sort of digging up these pits and pulling out these big blocks of sort of goethite, which I'll talk about in a minute. And they had to basically break them down. So they used a hammer and a chisel. This is all kind of the offcuts, I suppose. You know, and it's quite hard work. You sit there literally just cracking open these rocks from, you know, 7 in the morning till 5 o'clock in the afternoon. And the fossils are flattened. So they split when the rocks split open. And if you're lucky enough for a fossil to pop out, they're flattened. And they kind of split along the body plane. So the internal structures of these fossils get exposed. Now, as you can see here, there's soft tissue preserved. And interestingly, all of this soft tissue and also the matrix surrounding it is composed of goethite, which is an iron oxyhydroxide, which to me, when I first heard about it, I thought it was pretty weird. It's not the typical kind of sedimentary rock that you find fossils preserved. let alone exceptionally well-preserved fossils. I mean, as a geologist, I see goethite as a weathering mineral, not a sort of primary sedimentary feature. Now, the color you'll notice is a little bit different amongst the sort of insect in the backdrop of the matrix, and that's just due to crystal size variation, but we're still talking on a very, very sort of nanoparticulate size. Now, before I go into this sort of odd type of preservation, I just want to give you a bit more of a, exploration of the degree of preservation, the age, and the paleoenvironment of this site. So during just a couple of years, the team began to uncover an unusually high diversity and exceptional preservation of plant and animal fossils. They found a variety of leaf fossils, or plant fossils rather, that included 50 angiosperm leaf taxa, flowers, these are these little yellow flowers, they're probably about the size of my thumbnail, fruits, seeds, and there's also pollen and spores in this deposit, which I'll talk a little bit about in a minute. There's also a variety of insect fossils. So merolimnic insects, these are insects that have an aquatic life stage. That includes this list here that they've discovered. And just a few examples. So here's a midgie larvae. So those little, they're not mosquitoes, but everyone thinks they're mosquitoes. They don't bite. They're actually flies, but they're just tiny little flies. This is a larvae of one of those, so they're called phantom midge. So well preserved, in fact, you can see the internal structure of the contents of what was inside this little midgie. And if you look at the scale, I mean, they're tiny. They're this small, and that's the kind of preservation that we're getting. These features here are their little sort of raptorial bristles for them to catch their prey. Again, this is a dragonfly example. You see the internal structure of this dragonfly. They're beautifully articulated. Most of the fossils in this site are completely, the entire form of the fossil is preserved. They haven't been broken up in any way. There's terrestrial insects, so weevils, beetles, there's cicadas, really big cicadas, wasps and ants. So here's an example of a longhorn beetle and this parasitoid wasp. You can see these little sort of antennae here coming out, the segments of the wasp, just exceptional level of preservation. There are also spiders. Now, there's 13 taxa of spider that have currently been found, which apparently is 12 more than what Australia had. So prior to this, there was hardly any record of spiders in Australia. And so this site itself has expanded that record by what, 12-fold. This is a myglomorph spider. The legs are sort of cut off on the edges here, but this is kind of a predator, like a funnel web or something like that, quite a big sort of spider. Tanglewed spider, so there's a tiny little thing. They make sort of cobwebs, like three-dimensional cobwebs. And it's going to be hard to see, but on this little spider's leg, there are these filamentous structures you can just make out here, which resemble what could be the hairs on the spider's leg. That's the level of preservation we're talking about, and I'll go into more detail in a minute about that. It's exceptional. There is one dominant taxa of fish, just a little fish about this big, but we've brought out dozens of samples or specimens of those. They're stream type fish, and the contents of the stomach of this fish is also preserved. So you can see here these kind of vertical structures. Those are probably these little bristles from the phantom midge. So they were in this body of water eating these things, and those, what it was eating is actually preserved.
Can you just identify which images for the people online, because they won't see your red dot?
Oh, so point to it, or?
Oh, no, you just need to.
Oh, gotcha. Okay, so
say which.
What I got too many images on here. So on the basically the little bristles, if you look on the right-hand side, there's a box around the bristles on this little phantom midgie that's sort of sitting upright. And if you look just to the left below the fish, there are sort of filamentous or these vertically striated structures through the stomach. So you'll probably be able to see it pretty clearly on the screen when you're rewatching this. So this is just macroscopic preservation. We haven't even looked at things under the microscope yet, right? The microstructure of the fossil feather actually shows melanosomes. So these are pigment cells. Yeah, wow is a good response. So the image on the left-hand side here is an SEM of the actual pigment cells. So these melanosomes, and they kind of look like little, rice grains all stacked up vertically next to each other. The distribution and shape of these melanosomes indicates that the fossil feather may have been black or red. So, we're getting a lot of information from this site about the ecosystem. The fossil fish, same in the eye of the fossil fish. In this SEM image, we can actually see the melanosomes again. These are Omatidia. So on the right-hand side next to this crane fly, these little units, these little round units are actually the units of compound eyes in some insects. So like bees and stuff that have lots of little, those freakish little eyes, that's preserved. And stomata in the leaves, essentially. So, and just for a little scale, I put a little bacteria here. So these tiny little bacteria, that's sort of relatively in comparison for size, that's the kind of level of preservation we're getting in this deposit. And I've just put a little red blood cell here next to the stomata. So it is exceptional level of detail. I cannot stress enough how fantastic this find is. There's also microfossils. So this is a fungal spore on the the left-hand side here. You can see these are actually molds and casts of these organisms. And if you look closely at the matrix next to the spores and these yeast on the right-hand side, you can actually see the sort of detail of the crystals of goethite in the matrix. So, I mean, it is tiny. Like if you look at the, if you're looking at the scale bars where, you're hardly seeing any kind of crystal. This is nanocrystalline matrix of goethite. These yeast cells on the right-hand side, they actually preserve budding scars. So if you just look right in the middle of this image on the left-hand side, you'll see these kind of little pits all over the yeast. And you'll see sort of this thing at the top that looks a little bit like a nipple. That is essentially where the yeast split. So they're budding scars. That is, that's an exceptional level of detail. So I really want you to soak up how well preserved this site is. Possibly bacteria. Now we know this isn't sort of bacteria that's crawled in later and died, because in SEM images you would expect, say that it's sort of modern, something more recent that's come in, you would expect it still to be carbonaceous. And the rule of thumb with SEM is that things, the brightness sort of increases with atomic number. So this is all iron. This is all the color of the contrast of these SEM images about the same across it. We're not seeing sort of two different compositions. So these are probably indigenous microorganisms that were preserved during the fossilization process. And as I mentioned earlier, there are pollen and plant spores. So this is Anophthalgus pollen. There's a variety of pollen that's been preserved in this deposit. So of course, which is a bit weird because it's not the kind of usual way that you extract pollen from deposits, because these are being imaged on the surface of rocks. Because they're the molds and casts where, typically you would digest rocks to extract pollen from them. That's how it's traditionally done. In this case, it was definitely a pleasant surprise when they were first looking at these rocks to find pollen and spores on the surface of the rocks, and they were able to date the rocks using biostratigraphy. And here's the data of that. So basically, middle Miocene, 11 to 26 million years old, is the range that we get based on that pollen data, which is really interesting. And I'm going to talk a little bit later about, because of that surprising fact, another little bit of citizen science that came out of it. So in terms of the paleo environment, the dominant flora, we actually see things like climbing ferns, large leaves, not that you can sort of tell with that one, because I don't have any scales on here, but anyway, and drip tips. So this sort of the middle right image, if you look really closely, there's a leaf and it has sort of a, it thins towards the bottom of the picture. And that is a drip tip. So that's a wet environment. You're getting drip, that's where these drip tips form. Nothofagus, if anybody's heard of the southern beaches, these are trees that form in temperate environments today. And so the combination of flora and leaves and pollen that's found in this deposit indicate a mesic rainforest. So a moderately wet place, particularly during the growing season. Less abundant are flora of Metaceous and Casuarina type plants. So there's both leaves and pollen of these, but they are much, much less abundant. And this is more indicative of drier environments. So think, you know, modern day Australia, eucalypt forests. So you start to build up a bit of a picture. This is the interpretation of this site is an oxbow lake in a restricted mesic rainforest. So this is during the Miocene. This is, you know, when the continent was becoming more arid, drying out, and your rainforest is starting to be restricted to waterways. All of these finds are consistent with that interpretation. So you've got this beautiful fine-grained layered sedimentary rock, so nice low energy environment. As I talked about, the leaves and the pollen, the merolimnic insects, so it's sort of, they've got aquatic stage insects in this deposit. Terrestrial insects, stream fish are coming in, there are spiders, very high diversity. And one thing I didn't mention, we have thousands of specimens, and I'm about to talk about this in a minute, only one paper has been published on this, two papers I think been published on this site so far, and there's a lot of information to come out about the ecosystem that they've discovered. This is an artist's depiction, which is always nice to give a bit of a visual of what one might have thought it looked like, by an artist called Alex Boersma, who did an exceptional job, I think. So this work was recently published, only last year. It was a seminal work. The site is actually called McGraths Flat, and it was named after the farmer who found the first fossil, which is always nice. Now, not only do they describe the flora and fauna in this seminal work, but they also explain the significance, which I sort of touched on a little bit just previously. But the Miocene of Australia is a time when sort of mesic rainforest ecosystems that once dominated the landscape transformed into the shrublands, the grasslands, and the deserts of today. But there is really a lack of well-preserved fossils from this time. So McGraths Flat preserves a rare snapshot showing that shift from these ecosystems before the aridification of Australia. And it really is giving glimpses of the drying out process happening, right? We've got less abundant flora from sort of eucalypt type forests, and possibly, you know, this restricting of the rainforests. So this arguably gives us reason to find more of these well-preserved sites from this particular time that are similar to McGraths Flat. And how we do that, and where I come into this story, is we want to better understand how these fossils became well-preserved in the first place. So the previous work really did lay down the foundations for a taphonomic model, which is essentially the fossilization pathways. They documented the geology. They found that there is Eocene basalt flanking hills adjacent to the McGraths Flat deposit. And these are overlying some Jurassic sandstones, of which contain those Talbrega fossil fish beds. In this model, we have basalt being in place during the Eocene. And then during the Miocene, iron is leached from weathering basalt, so turning to saprolite perhaps. And this iron is entering the lakes, the rivers and the streams within a paleo valley. The ferrous iron is undergoing some type of oxidation to ferric iron and producing goethite, perhaps during rainfall events, which is bringing oxygenated waters, perhaps during evaporation. We're not sure yet. And this precipitated fine layers of goethite in low energy water body, which has encased and replaced our organisms. This is present day McGraths Flat. The erosion and weathering is sort of, left the topography perhaps somewhat similar to maybe what it was before, but maybe the valley was more incised in the past because the deposits are actually flat lying. There's no dipping, and certainly not in this area. Now, we need to test this hypothesis. So this is where it gets fun for me. We also have a lot of unanswered questions. So as I mentioned, I kind of joined the team in about 2020 as a part-time research assistant. And during that time, because I have a background in geology, I got interested in the idea of looking at the taphonomy of this site. And so Matthew McCurry, who is the current curator of paleontology at the Australian Museum, suggested we can put in a linkage grant, which is what we did, and which is why I'm standing here now. And we came up with this integrated approach. It's an extremely multidisciplinary approach. And today, admittedly, we have more questions than we do answers because I've only really started to sink my teeth in 2 weeks ago. So get me back in 2 years and I'll have 80%, maybe. All right, so first off, can we constrain the age of McGraths Flat? So through biostratigraphy, we know it's about 11 to 16 million years old, but you can actually date the goethite, which is really cool. I didn't know that you could do that, but you can use uranium, thorium, helium geochronology, where basically they're measuring the accumulation of helium-4 particles, and that can give an age of goethite, which is pretty cool. Now, I think there's a couple of different sort of aspects or answers to questions that we have. One, we can possibly constrain the timing, obviously, and perhaps the fossilization pathways. Also rule out the fact that the goethite may not be primary, although I would argue that level of preservation, getting melanosomes and cellular data, I do not know. That's a great place to get reverb. That's all right. Yeah, I do not know how you would get a deposit with this level of preservation through the process of altering from some other iron phase to goethite. I cannot see that happening. But it would, doing this geochronology at least would give us an indication that that goethite is forming during the time by which the ecosystem is being deposited. Possibly time averaging, depends what the error is that we're getting, obviously. But, maybe, so this deposit is, 30 to 50 centimeters thick. And so we can take analysis up section and try to somewhat understand whether this is a long duration of deposition, a short duration of deposition. Again, that really depends on what level of error we're getting in terms of the geochronology. If it's sort of, multiple events or continuous deposition, from a sedimentological point of view, it certainly looks continuous. And even from sort of ecosystem, I mean, you're not seeing a huge amount of changes in terms of the flora and fauna. There's not like evolution happening in the time that this thing's being deposited, right? Now, I promised a side of citizen science, seeing as we're talking about dating. One of the sort of offshoots from the fact that these pollen are able to be observed on the surface of rocks spawned a citizen science project, because typically, as I mentioned earlier, we digest the rocks, and that's how they extract all of the pollen. But in this case, it appears to be, one of the first times that, well, certainly I know of, maybe it's not the first time ever, but to actually image pollen on the surface of rocks. This came as a surprise to the people that were looking at these rocks. And obviously we can age date and we can get environmental reconstruction information. So, which is great, but when you're doing it as a single person on an SEM, if anybody's ever done microscopy and had to do a lot of microscopy, it's a very arduous task. And so there's some limitations in doing this. It's time consuming, it's costly, there's observation bias. Big pollen as opposed to small pollen are going to be the ones that are picked out. Obscure pollen are going to be missed. And it's qualitative only. So this idea was struck. The Centre for Advanced Microscopy at ANU has a system where you can automatically get SEM images and essentially make a mosaic of 3,600 images across the surface of a rock. So what they did is they got 7 of these. The math's wrong, it's 25,200 high resolution images across an area of about 1.85 centimeters square from 3 different samples. So it's not just one big chunk of area, it's from a couple different samples. And they took all of these images and they invited citizen scientists to browse the images and help us ID the pollen. Now, I've skipped a lot of the methodology here because, you know, time doesn't sort of permit me to go through 2 completely different projects. So I've condensed it, but basically, we invited these citizen scientists to browse the images. We did this through a fully online platform. So these citizen scientists could essentially log on. They could, you know, go through answering a bunch of questions. We give them an ID manual and they could help us ID if anything was in the image. This was done on Digivol, which is a platform through the Australian Museum in collaboration with the Atlas of Living Australia. I think funded also by CSIRO. It's a great citizen science platform. Australian Museum has a number of different citizen science projects, including Frog ID, where people can go out and, you know, use their phones to see what frogs are out there based on the sound. It's great. It's an excellent platform. So we got about 271 citizen scientists to help us do this. We had this data fossil project up for about 3-4 months. And so in terms of the limitations, it kind of solved the problem of, you know, the gentleman actually the collaborator of ours who did all the SEM work, Michael Frese, saved him from sitting at the SEM for hours on end. And it's cost effective and we have more eyes observing the data. So we're not missing stuff across the surface of the rocks. And it increases public involvement in paleontology, which historically is actually quite difficult, particularly because fossils are fragile. Getting out into the field is hard. You know, this really gives an opportunity for people to be part of the process of science, of paleontology in a way that probably has not been able to be done before. Technology. So results. So again, I have condensed this and it's only a 5-page paper that we published. So if you're interested in more detail, I can point you to it. But basically what we did is we were able to reduce that 25,200 image data set to 4,000 images. So 21,000 images had nothing in them. That saved me from looking through 21,000 images. We had a step-by-step sort of validity process to make sure that, you know, that wasn't the case. We weren't missing anything. Again, detailed in the paper, and I won't sort of bore you with the details here, but really, you know, the key take home is 20,000 images we did not have to look through because they would have been empty. That left us with 4,000 images to check. Out of those, about 10%, so about 450 odd images had something in them. And there were 300 pollen and spores that we were able to identify. We had an expert palynologist check our identifications. And of that data, we can in fact determine the age and the environmental characteristics of our fossil site. So this method works. This is some of the data that came out of it. So this is our 300 pollen. We can also sort of say something about their distribution because we got it from a set area. And, you know, most of these, except for 2, we actually found 2 new taxa from this process that had not been discovered before. So back to what I was talking about originally. Did iron, so next sort of thing on the list, did iron originate from the tertiary basalt? So here's where I just have a bunch of questions for the rest of the talk. In this case, we're going to try and look at the trace element geochemistry. So if iron is being liberated from the basalt, you would expect other elements to be liberated as well. So perhaps we can compare the composition of the basalt to McGraths Flat rocks to try and link the two. There is limitations with this. Iron oxides are known to influence trace element partitioning. Unlike carbonates, like seawater carbonates will have a typical rare earth element signature because of the inventory of elements they come from. You know, it's very easy to sort of figure that out, but in this case, it could be a little bit more tricky. But if we can sort of determine that, yes, this is where it comes from, then it does have implications for locating other goethite-rich Miocene fossil sites from the point of view of geological context. Do we look for sites that are associated? Do we look for goethite sites that are associated with basalt? Again, gets a little bit tricky because you would expect to find possibly some laterite or something like that along with basalts. But questions, right? We've got to start somewhere. What were the environmental conditions? So trying to constrain the local environmental conditions a little bit more, we want to look into water temperature. So looking at oxygen isotopes, water depth. So there's an aspect of looking at the aquatic insect assemblage and compiling data of all of the different sort of aquatic insects or life stages of the aquatic insects and seeing if there's any sort of, you know, linking features of what type of water depth they might be living in. Chemistry, you know, is there a chemocline? Is there an oxygenic bottom waters, for example, you know, bringing in sort of free iron to then be precipitated? You would expect there must be some sort of dynamic chemistry happening. There's no calcifying organisms, there's no bivalves, there's no gastropods, there's no shelly sort of organisms. There's no little Daphnia, so there's these little water fleas that are eaten by the midges. We don't see any of those, so they prey on water fleas. We don't see any of those, and they are essentially planktonic crustaceans. So what's happening to those? Maybe they didn't feed on them, who knows, but it's potentially says something about the chemistry. The only calcifying organism we do see is something called a glaucidium, which is a little bivalve, so a little sort of shelly organism larvae that's attached to one of the stream fish. So typically these bivalves, they actually send out, you know, to send their little offspring off to have a good life. The larvae will attach to a fish, the fish will swim away, and then they'll, you know, spawn, so they'll grow somewhere else. We do have one that's attached to a fish, which is preserved. So what that says about the chemistry, you know, potentially it's not to the point at which that sort of is dissolving. All right, so what about the precipitation of goethite? What are there abiotic implications or biotic implications in terms of precipitating the goethite? Now, interestingly, in Picton, I used to do some sort of hydrogeology monitoring of a coal mine down there, and at the time I was partly working on McGraths Flat, and I saw this in the creek bed, and these are leaves that are being coated by iron oxyhydroxide, and I've been told that this is due to microbial mediation. So the precipitation of goethite through microbial mediation in these pools. Now these are thin films. In a storm, which happened all the time, that would all just wash away. So how do we get nice big thick deposits of this stuff forming? So it must be, you know, a relatively deeper type of environment to be able to form this kind of thick deposit. So are Miocene conditions conducive to this? Is it something about that time and that environment that's actually conducive to the formation of these thick deposits? We want to investigate more modern analogues that might be a little bit more similar to the conditions that we think that this deposit formed. So as I mentioned earlier, sort of abiotic precipitation of goethite has been suggested. So rainfall coming in and oxygenating the water. But microbial mediation is definitely a possibility. And in fact, if we have a look at this image I showed a little earlier, there is this cavity, the semi-empty space where possibly organic matter was previously. And these little microbes, so are we possibly seeing bacteria preserved decomposing the organic matter? Which would be pretty cool if that were a correct interpretation. That's it. I think that a shout out to Malcolm Walter if he listens to this, because I sat down and showed him some of these images, and he's an expert in this area. This is quite interesting. So in terms of sort of stages of fossilization, we actually have images from a instrument called MyMapper. Chris Ryan from CSIRO in Melbourne helped us get these. So this is a high definition micro XRF element mapping system. And basically, so it's about 30 microns resolution. As you can see, these arrows are pointing essentially in the direction of where iron is being concentrated, and they're both pointing at some fossils. So the red is titanium, the green is iron, and iron is being concentrated in these fossils, which is also another good cause to believe that iron in this system is actually an early process. So why is it preferentially going into the fossils unless the fossils have something that is attracting the iron, which is probably organic matter? We know things like pyrite adsorb onto fossils. So why not another phase of iron? Other compositional controls on preservation. So in, sort of on the back of what I'm just talking about, is there any sort of chemical or textural features of the fossils that might be controlling the sort of preservation style? Because we do see these different, so these colors are all to do, I mentioned earlier, to do with the crystal size of the goethite. It's all goethite. It's just presenting, it's manifesting differently. So black is generally sort of amorphous or cryptocrystalline, I guess, in a sense. I mean, this is already pretty much that. Yellow is increasing in size. We do get sort of little tiny veins of amorphous or vitreous black goethite, which is probably a late stage diagenetic effect. But there's definitely something sort of going on in terms of preferential or controls on this composition or manifestation, at least, of the goethite, because some examples include fish. They always seem to be yellow. Insects always kind of seem to be these dark reds and browns. Internal organs are often sort of yellow. Leaves sometimes appear greenish yellow, which is a bit weird because it's definitely not the original leaf, but it does look that way. But anyway, it'd be interesting to sort of understand what are the controls in terms of goethite precipitation in these areas and why. Up in Queensland, we have a collaborator, Paulo Vasconcelos, who has we're going to be going to do LAICPMS, so trace element mapping across some of these samples to see if we can't decipher if there's any elemental concentrations that give us a bit of indication to answer these questions. And very last thing, what geological features control this iron-rich Lagerstätte? So can we devise some sort of search strategy to locate other information-rich fossil sites in Australia? Do we look for goethite deposits proximal to tertiary basalts? That's pretty vague. But still a question to be asked. Were goethite deposits widespread in the Miocene? If they were and they're preserved, then perhaps we should start to look for these types of deposits, these iron-rich sort of horizons. And what other geological characteristics could we use to locate such deposits? So these are some of the questions that we really don't have any answers to. If anybody has any ideas, I'm certainly open to hearing them. Just as some preliminary conclusions to wrap it all up, what we do know and what we want to know. I'm pretty confident that the goethite is at least an early sort of part of that fossilization process. As I've shown you with all, I spent a lot of time showing you the exceptional level of preservation of detail for a reason. And I think that really backs that claim up. But we want to understand, so, you know, the different stages that are involved in fossilization of this, this deposit and determine some of those controls. Modern examples of goethite do exist. I've seen them sort of coating fossils, but how do we get these nice, big, thick deposits? Perhaps the Miocene epoch may have created the conditions to form these goethite deposits. And recently, just last week, I was reading about channel iron deposits in the Hamersley province that do have these sort of thick deposits of goethite, but rather than being this beautifully fine-grained goethite that I'm showing with preservation of, you know, exceptional detail, they're pisolytic sort of gravelly things. They're mined for iron ore. That's, you know, what the, you know, how they end up in the literature. They do have some fossil wood in them, so it's not the kind of preservation we're potentially looking for, but they are Miocene age, and the formation of them is somewhat similar in terms of this sort of taphonomic model. So it does give, you know, it seems sort of logical that maybe this is a time in Earth's history or Australia's fossil record that this is the kind of deposits we should be looking for. And again, which I just mentioned, constraining these details and potential model for exploration, and citizen science. It's great. It's an efficient method to help date and quantitatively assess these environmental characteristics in deposits that cannot be digested. So not just goethite deposits, which would be great because we can use that if we do find more, but also other deposits that, you know, may have information that you don't want to sit at a microscope for hours and hours, and you can automatically gain a huge image data set to, and then have citizen scientists get in the process of, you know, doing science and contributing to that, and then, you know, making that process a little more efficient, and involving the public in paleontology, which is always a yay for me. And that's it. I've got a couple of acknowledgements though. So this was the data fossil team. Really collaborative, or sorry, diverse team. And the McGraths Flat team is composed of a lot of different people. There definitely needs to be some shout outs to the volunteers. So these are longtime volunteers. Robert Beattie, his love of insects, I'm told, is partly, it should be recognized partly as like why they kept going back to this site. This new site to check it out, because he's just an avid fossil hunter. Matthew McCurry is the curator of paleontology at the Australian Museum, and he's done an extremely good job of bringing together a diverse group of people to, you know, make this work actually happen. So really a huge shout out to that. And a lot of people are involved. It's a very broad expertise of group. There's a lot of volunteers involved. We couldn't do this without all of these people. So it's worth mentioning all of that. Very last, the Etheridge family descendants. So Robert Etheridge, who was the first curator of the paleontology collection, I believe he started in 1887, 100 years before I was born. His descendants kindly funded some of the work. The Australian Research Council and UNSW, volunteer citizen scientists, I really cannot stress enough, like they put their huge amount of time and effort into doing this sort of work for free. The Australian Museum Research Institute and the Australian Museum Trust, who's supported this project. Nigel McGrath, who always kindly allows us to access his property to look at these fossils. And I want to acknowledge the custodians of the land and waterways on which McGraths Flat deposit is also located, which is the Wiradjuri Nation people. And I think that's it.
Okay, great. Thanks, Tara.
Cool. I'll add one last thing, which is a bit of a job ad that's come up at the Australian Museum, which I was exclusively asked to mention while I was here. There's a TO position in the Mineralogy Department. So if you have any students or colleagues that are interested, there's a 18-month contract. So yeah, they don't come up very often. If anybody wants the details on that, I'm happy to send you through the link. Cool. Now I'm happy to answer any questions.
Great. We'll only have time for a few questions. Tara tells me that the restaurant's closed.
So we need to get down there to order.
Oh.
Or they say they're not taking any more.
Which is when?
They actually close at 8 o'clock.
You don't, yeah, you can skip.
So we've
It's
got,
25 to.
we've got a question, let's take two questions. We've got a question from Mike Smith, who says, fabulous images, Tara. Question, has silica precipitation contributed at all to preservation of microfossil plants?
Not, there is a little bit of silicon in the deposit, but the XRD analysis doesn't show anything but goethite. So it's a good question, and certainly, you know, worth considering what else is in there. But at this point, there's no good cause to believe that goethite isn't the thing that is causing this sort of preservation. Yeah.
Great. The Maya images show up silicon?
The Maya images, I don't have the data to answer that question, because the only data I have is on titanium and iron at this point. But yes, theoretically, if there was, theoretically, if I had access to it, I could answer that question. But there's definitely a bit of silicon in there. There's a little bit of calcium, there's a little bit of sulfur. So and I didn't want to go into it in too much detail, because there's sort of questions that I have which I want to explore in a bit more detail from that aspect, but definitely worth asking those questions and having a look into it a bit further, for sure.
I suggest everyone who's here will ask lots of questions over dinner. So everyone should join us for dinner downstairs. And so yeah, please head to the front of the kitchen and order immediately. Okay, thanks so much, Tara.
Cool. I know. It's insane. Yeah, I know.